So, can you see my screen all okay?
Yes, we can.
Right, so I'll get started. So, thank you, Alison, for that introduction. So first, I'd like you to imagine if instead of COVID tests, we could have just smelled when someone else was infected. Well, that might actually be possible in honeybees, and my PhD set out to try and figure out whether bees are capable of this. So, my talk today will cover these topics here. So first, we'll cover disease detection and social immunity in honeybees, with this idea of social immunity. And then we'll move into some of the experiments I started doing during my PhD. So looking at what we already know about infection smells in bees. Can we actually detect infection in honeybees? Then going into some behavioral studies where we tried this in the lab to see whether bees can use these smells to detect the infection and maybe avoid infection, and then scaling that up to whether they can do it in colonies as well. So I'm preaching to the choir here, of course, but honeybees are incredibly important parts of ecosystems, providing us with a whole range of ecosystem services, but mainly through their pollination services. Almost half of the leading global food commodities depend on animal pollination in some way. And that value is estimated in terms of the value for that comes from human food, is estimated at about 132 billion pounds a year. And honeybees are obviously incredibly important parts of this process. Wild pollinators are equally as important, but in agricultural settings where we have big old monocultures, the ability of honeybees being incredible generalists with really large foraging ranges is what makes them particularly suitable in these kind of agro ecosystems. And of course, alongside that, we get honey, and that's estimate about £7 billion a year. So both its ecological and ecosystem services and the financial benefits that they sow. That's why honeybees are so important to us. But they do face a wide range of threats. Since 1961, the demand for pollination has grown by about 300% as the population has expanded. But the honeybee stocks in the world have only increased by about 45%. We think this is due to the high levels of mortality that honeybee colonies face. So this is from a survey in America from 2008 to 2022. If I whip out the laser pointer here. So the kind of acceptable rate of what you would normally expect of colony rate loss is around 10%. And you can see since these records began, we haven't really even come close. And it's thought that there's a wide range of reasons why this is, and a range of environmental stresses, but the two real big ones are pesticides and parasites, which can often interact with each other. So my PhD, of course, focused on the parasite aspect of this. So as beekeepers, you'll all be aware, and especially if you're a bee inspector, of the many, many diseases that bees can face. These are quite various. We have bacterial infections like American and European foul brood, particularly nasty infections, notifiable, really transmissible, and hard to get rid of. We have things like Nosema, which has now been reclassified to be called Variomorpha, these fungal microsporidian gut parasites of adults, which can cause colony collapse sometimes and weaken the colonies. Varroamite, the worst of the lot, really, in terms of its ubiquitous distribution around the globe and its ability to vector a range of viruses, such as deformed wing virus, that causes adult bees to emerge with deformities and they end up dying. And then things like chalk brood, where kind of these fungal infections of the larvae end up forming these mummy-like structures and killing them. But how do honeybees naturally defend against this wide range of threat? So there's this idea of social immunity. And all throughout the animal kingdom, not just in honeybees, there's been this idea of, this evolutionary process of behavioral avoidance of infection. So while we as mammals have antibodies and different immune responses, really the best way to avoid infection is just to not get infected in the first place. And so we see examples of this throughout the animal kingdom through to mandrills, birds, bats, lobsters, bullfrogs. They all show some behaviors where they have evidence that they might be avoiding individuals that are infected. But in social insects, this is particularly important. These are highly related, very dense colonies of individuals where disease can rapidly transmit. And honeybees in particular rely heavily on these collective behavioral defenses. And so if we think about the immunity genes that insects encode within their bodies, these would be things like innate immune response. If we look at non-social insects like mosquitoes and fruit flies, honeybees have about a third of the immunity genes as those kind of insects. And that means that over time, through evolution, they've invested very heavily in the more behavioral side rather than kind of the innate immunity side of things. And so social immunity acts at all potential stages of infection, reducing the parasite uptake and intake. So an example of this is honeybee guards. Sometimes they won't let back foragers that are carrying infection. So it stops that parasite being taken into the colony. Stopping the establishment in the nest. We see this in termites, ants, and honeybees, where we have dedicated kind of, well, in ants in particular, they have dedicated graveyards for where the dead are put, and honeybees will remove the dead from the colony to stop those infections establishing. And then preventing the spread within the colony. So there's a really interesting paper that shows that bees with varroa mites will socially distance away from the brood areas so that they can prevent the varroa kind of moving towards those more sensitive areas of the hive in some cases. But how does a bee actually know who is infected? It's not like bees are going around sneezing and coughing, so there must be a way that they know when the other individual is infected. So chemical cues and olfaction, and this idea of smell, is really important in many, many insects. It's actually one of their fundamental senses. They communicate using pheromones and chemicals in the air and on the bodies. And there's two main types of chemical that we kind of talk about in this context. This idea of cuticular hydrocarbons. These are waxy substances that are found on the surface of insects and other invertebrates. And they tend to be detected using the antenna. So a bee will walk up and they'll antenate each other and they're detecting these cuticular hydrocarbons, particularly in Hymenoptera like wasps, bees, and ants. These are really useful for nestmate recognition, so knowing who is part of your nest. And then you have volatile organic compounds. So these are things in the air. Think of things like queen pheromone and the alarm pheromone, long-range signals that bees can detect as it moves around the colony. And there's loads of evidence that cuticular hydrocarbons do change when a bee is infected, and that's detectable. So in bacteria, such as American foulbrood and European foulbrood infections, varroa mite infections, a range of the viruses, fungal infections as well. But there's far fewer studies that have looked at the volatile organic compounds, these long-range communication signals in bees. So another really interesting side of using these long-range communication signals is that they could potentially be used to monitor infection as well. If a bee can smell it, maybe we can smell it. And so if you think of kind of like a carbon monoxide sensor for your in your home, you could think of that as like an infection sensor inside of your colony. So I published a paper talking about these kind of things and using this in insect agriculture. The idea being that maybe when infection arises, the sensor could ping something to your phone and tell you when infection's popped up in the future. We're not quite there yet. But another really interesting part of this is because volatiles are so intrinsically linked to the metabolism of the pathogens, it might let you smell it before you see it. So you could treat the diseases earlier before they get too advanced. So my thesis set out with these key questions and goals that we were aiming to answer. So to what extent are volatiles associated with honeybee infections? And is this just in the brood or is it in the adults as well? When in infection, can we detect those volatiles? So this would kind of go back to, can we detect it before the visible symptoms arise? And finally, are bees actually using this to inform on their behaviors? So my first experiment that we set out to do is to find out what we actually already know about volatiles in honeybees in association with infection. So there's a few studies out there that have previously linked infection with changes in volatiles. Several of the major honeybee diseases have been investigated, things like Varroa and American foulbrood, but the findings are scattered across lots of individual studies. And so what we set out to do is to bring all of those together and do one overall synthesis to see if there was any patterns that were emerging. And so what we did is I screened through over 1,000 published studies, and I filtered those down to 23 key studies that just looked at the volatiles associated with a range of different infections, or whether the bees were uninfected. And we also included the parasites on their own. So in vitro, meaning maybe like a mite on its own, or chalk brood that's grown in a petri dish, rather than actively infecting a bee. And we were looking at the presence or absence of different chemicals that they were emitting. So we didn't include in this study how much of each of these individual chemicals were, just whether they emitted them or not. And the idea being that we could pick up these broad patterns, and maybe there was certain patterns that are associated with different infections that could be useful for understanding how we might monitor them or how bees might use them. So to do this, to make sense of these really complicated data sets, there's a type of analysis you can do called network analysis. And I'll briefly describe this before I show you the data, just so it makes a little bit more sense. And so with network analysis, in this context, we can treat each volatile, so each of the chemicals that we found in these studies, as a node. And they would be connected with another volatile if they're both emitted by the same parasite. And then the strength of these connections can increase if there's more more treatments or parasites or the uninfected bees that also emit those compounds. And the idea being that the more common things will be highly connected in the middle here. And then we'll get these clusters on the edge that are less common, more specific to certain parasites and infections, and give us an idea of which volatiles are associated with those conditions. And the idea being here is that over time, once we have done this analysis, we can kind of identify these scent bouquets of disease. And so this is the network that we constructed. We found 279 volatiles that have been published across all of these studies. And you can see using these algorithms that we've kind of passed the data through, they've clustered them into 8 different, what we would call communities or clusters of smell. Several of these communities were strongly linked with disease, and we'll dig more into that in a second. But you can see these 8 communities here, there's kind of 2 big ones, and they've managed to get these clusters on the outside that the algorithms have decided that they're way more connected with each other than they are with other communities, and so they need to be separated off. So the smell of infection is real, but it is quite complicated. And so we can look at the identity of what is inside these clusters. So remember, again, these nodes here are the volatile compounds, and then the links between them are the different treatments that they're associated with. And you can see in these 2 communities, these really big ones here, these are all associated with a wide range of different treatments. So different parasites, uninfected bees, the mites in vitro, so mites on their own, mites infecting bees, kind of general smells that are associated with bees. When it gets really interesting, if we look into these communities here, this is a whole range of volatile compounds that have only ever been reported across all of these studies as being associated with American foul brood. The same goes here for a few of the chalk brood treatments. So just highlighting these ones here, we have some that are associated with in vitro chalk brood, some that are associated with chalk brood when it's actively infecting a larvae. And then one of these, there's only emitted by, well, that is emitted by both of them together, but it's not ever emitted by any of these other treatments. And so if we actually look at the identity of some of these, and there's a couple of communities also that are associated with varroa and in vitro varroa as well. So when we start to look at the identity of what compounds are in these different communities, so if we remember community 1 and 2 was this big mixed group, and these were compounds that are associated with normal functioning of bees. So sometimes there would be pheromones that the larvae use to communicate with the adults. And so these would be kind of typical bee smells. Even if you're infected, you're still going to produce some volatiles that a bee produces, just, because a bee smells like a bee at the end of the day. In community 3, that one that was associated just with American foul brood, these were all associated with stress. So pheromones we know bees produce when they're stressed, and also decay. So a lot of sulfates that are associated with decay, which might explain that kind of weird, horrible smell that American foulbrood produces. In the community that was associated with just chalkbrood that was grown on a petri dish, we saw fermentation compounds that associate with fungal growth. So these volatiles could be associated with the metabolism of the fungi itself. And then the ones that are associated with chalkbrood, when it's growing on the bee itself, were associated with a couple of different pheromones that we know trigger hygienic behavior. So we might think here that the bees are producing these pheromones to signal for themselves to be removed from the colony. And then in communities 6 and 7, we have these bee pheromones that are associated with stress, again, hygienic behavior triggers, and also some that are specific just to the mites. And these could be quite interesting from the perspective of quantifying how many mites are in your colony without having to open up the colony or do any quantifications manually yourself. And then we had that community where it was just one volatile, has no known functions, but it was emitted both by a chalk brood that was grown in vitro and chalk brood that's actively growing in a larvae. This one could be really interesting, but it did only come from one study. And so I would be skeptical as to whether it might be a contaminant that they picked up, and my analysis has pulled it out as something more unique than it is. But if it's not, it could be a really interesting, different biomarker of infection that could tell you when you have chalk brood. So why does this matter? Well, if infections do produce distinct scent bouquets, then we can use this to train gas sensors that could allow you to monitor infection non-invasively. It could also be that bees are using these volatiles to detect and respond to disease, but also the parasites themselves could be manipulating the bees. And so parasites in the past have been shown to manipulate the smell of bees to make them more transmissible to other colonies. So from an evolutionary perspective, it could be quite interesting as well. But I think one of the key things that this study pulled out is you might have noticed that Nosema or Varomorpha wasn't included here. And that's because no study in the past had looked at the volatiles associated with adult infections. They all looked at brood infections. And that struck me as kind of weird because adults are also really important parts of how disease moves around a colony. They're the ones that go out onto the flowers and pick up parasites and come back. And so the fact that nobody had looked at the volatiles of adults in associated with infection struck me as strange. And so that's what we set out to look at next. And so this experiment here is one of my favorite titles of a study that I've ever managed to get through the reviewer process is Wake Up and Smell the Infected Bees. And so what we set out to do here is look at the volatiles of infected adults. So like I said, no studies have done this before. And to do this, we looked specifically at Varomorpha because this is a very common parasite that you get in colonies, quite ubiquitous across the globe. And we asked whether the infected adults would produce detectable volatile signatures. And then we looked at this throughout time. So this goes back to that key question of when during infection can we detect the difference in volatiles? And so for those of you that don't know the life cycle of Varomorpha, what tends to happen is a bee will go out and forage on a flower. They'll pick up spores if they've been deposited on there. As they ingest them, they come back to the colony and it starts to develop in their gut. As bees share food in the colony, they transmit between the different workers. They develop in those workers, and then the forager leaves again, starts depositing on flowers, and the cycle repeats. So it's associated with colony weakening and stress. So it's not one of the worst diseases, but it is quite important to control it. And it can alter the bees' physiology and their gut microbes, which are also very important to bee health. And so this was kind of the experimental design here. So I would take bees back to lab, we'd rear them in the lab, and then I would experimentally infect them with Varomorpha spores that I had isolated. And then I would sample their volatiles at day 0, 3, 6, 9, 12, and 14. So as the infection progressed, we'd get samples to understand how their volatiles change over time. But to do this, I had to figure out how you actually collect the smell from living bees, because I didn't want the smell of dead bees. I wanted this to be usable in a real life situation and be specific to bees when they were alive but infected. And so I came up with this method where we would keep bees in these sealed enclosures and we'd rear them in there and we'd give them food through a feeder. And then I would have a sampling port that connects directly to the inside of the cage. And then you can see the actual process here. We would use a special tube that we can then pull the air out of after the volatiles have accumulated in there for a while. We slowly draw out all of the head space, we call it, so the volatiles in the bag. And then they get adsorbed onto this tube, and then we can run it through what's called gas chromatography mass spec, but you don't need to worry about that. It's basically funky machinery that can tell us what volatiles are in that sample. And so, again, to make sense of this data, we found 71 volatiles in total across both the infected groups and the uninfected groups, but none of them were specifically unique to either treatment. So what would happen is that the concentrations of volatiles that are emitted in both uninfected and infected bees would vary slightly. And so you need to use special kinds of analysis that can pull these trends apart. So one of these is called ordination. And so ordination essentially, as my very in-depth graph here shows, let's imagine these bees only have two volatiles they've ever emitted. And this is the amount in nanograms of how much they're emitting. So you can see that this bee here is more or less similar to this bee here. You know, the volatile one, 8 nanograms, volatile one, 10 nanograms, but very different to this bee down here that hasn't emitted hardly any of them. And so what the analysis does is it gives you points that can move around this space. And as the different samples that are more similar to each other will cluster together, and the ones that are more different will move apart from each other. And so that's what we did with our samples. And so this is what it looks like. So remember again, each point here is a replica of the cage of bees that have been sampled with the smell, and their position is changing and moving around based on the different levels of volatiles they're emitting. And so we can think of this as an overall volatile profile. And you can see at days 0 and 3, they're very similar between infected and uninfected bees, which is what you would expect because the infection hasn't progressed much. And we start to see a difference here at day 6. So there's quite a lot of spores being produced here. The bees are starting to get quite infected. And we can see that their volatile profiles are different. They're no longer overlapping. Then strangely, at day 9, they become very similar again. And then day 12, very different. And then day 14, they became similar again. A very weird process to start with before you start to dig into it, because you would imagine that at day 6, they start getting different. And then over time, they get more and more different or they stay different. But this kind of fluctuation between similar and then dissimilar was quite strange to me. And so to understand this, we looked into how Variomorpha or Nosema actually develops inside of the gut. And so to do this, I had to pull together two different studies because nobody had sampled past. So this study did between days 0 and 12, and this study did 10 to 20. Nobody did the whole range. So we had to pull together these different studies. So if we look between days 0 and 3, there's not really much replication of spores. So this is the number of spores along the y-axis here, and then this is time along the x-axis. But when we get to day 6, this is when we first saw our difference. Just after day 6, there's a massive increase in the number of spores. And this could signal that the Variomorpha inside of the gut is changing its kind of genetics and its growth factors to get ready to rapidly replicate. This is something we know many parasites do. They go through these phases where they change their genetics and their metabolism through different phases of growth. And if we go to day 9, when again the difference kind of comes back to being similar, there's this plateau where there's no longer this rapid growth anymore. If we go back up to day 12, when it's very different again, it's right in the middle of this rapid growth stage between 10 and 14. And then at 14, it plateaus again. So what we think could be happening here is that the volatiles are changing, becoming very different as they go through these different growth stages, as they're kind of rapidly replicating, which could be quite interesting because it's directly then linked to the metabolism of of Nosema itself. So what does this mean? Well, we've shown that infection does alter the volatile profiles of adult bees at specific times, and we can detect that by day 6, which could be quite important. Day 6 isn't the peak of the infection, so it is quite early in the infection's progression. So, from a disease surveillance perspective, if you could pick this up in the colony itself, you could then apply earlier treatments. This does then answer our question from before that the volatile changes, as we showed in our previous experiment, happen in brood, but they also happen in the adults. So we go back to the key questions of my thesis. We've answered this question. Yes, the volatiles do change with life stage, and it's specific to certain stages of infection. But something interesting from this is that the changes in volatiles happen at this overall profile level. There weren't any unique biomarkers. So if we're thinking about monitoring this in an actual colony, it becomes a little bit trickier because there's nothing specific just to that infection that we're finding. But it is possible. Some people have managed to do it, but it does get a bit trickier. And it means that gas sensors, if they could be trained to these profile changes, could potentially detect infection by day 6. But there leaves one more important question here, is whether the bees themselves can actually detect these differences. There's nothing to say that these volatiles change, but it means nothing to the bees. So we have to start looking at some behavioral experiments. And so that's what I set out to do next, to understand whether bees actually respond to the smell of infection. So we started off simple in the lab, looking at just pairs of bees, and they would either be paired together with one infected bee in one side and one uninfected bee in the other side. And then as our control comparison, we would have two uninfected bees. So uninfected bee, uninfected bee. To try and isolate volatiles as the signal that they're using, I created these arenas here where some of them would allow direct contact. So the bees could antenate each other and detect both the volatiles and cuticular hydrocarbons. Whereas in these touch prevented arenas, only the volatiles were allowed to get through. They couldn't physically antenate each other to detect those cuticular hydrocarbons, so the less volatile compounds. I then looked at how often the bees would come near each other to see if they would be socially distancing from each other, how often they contacted each other. And also, I had the idea that if an uninfected bee was paired with an infected bee, would they auto groom themselves more? Because you see this throughout nature. Other invertebrates will groom themselves more in the presence of infection, as it's obviously a natural response to reduce the levels of infection on their bodies. And so we measured these behaviors. And the idea here, again, is trying to isolate whether it's volatiles or if it's the cuticular hydrocarbons that really trigger the differences in these behaviors. And so to do this, I set all of my arenas up. So we do the same infection protocol as we did before. And we were doing this with bees at day 7. So if you remember, at day 6 is when we started to get the difference in volatile profiles. So we know that they should start smelling a bit different at this stage. And we use this fancy software to record the movement of the bees throughout a 2-hour period. And in total, we had about 415 hours of video per pair. And this would allow us to then calculate how often they contacted each other, how close together they were throughout the entire recording period. But then to actually look at the auto grooming, the software wasn't fancy enough to be able to tell us which behaviors they were doing. So I had to manually go through this 415 hours of footage and then look at all the different auto grooming behaviors. So I would count. So every 2 minutes, I would watch the footage for 30 seconds and I would count how often they were auto grooming. So this was the data. And surprisingly, and sadly enough in science, this does happen often. We didn't really find any differences. But no differences does not mean a negative result. It just tells us a different answer. So what we found is that the contact rates between them wasn't really different at all in either arena. So even if they were two uninfected bees paired together, or if the bees were paired with infected bees, it wasn't too different between any of the conditions. There's slight difference here, but you can see these bars show that the variation was very high. And so when we did our analysis, it wasn't statistically different. And the same with auto-grooming frequency. So none of the bees in any of the conditions groomed themselves any differently, regardless of who they were paired with, whether they could only detect the volatiles or whether they could touch each other. And so we tried to understand why we were failing to find a response. Previous studies have shown in laboratory settings that if you introduce bees to other bees and they are infected, they can be attacked and avoided more often. And so it seemed to contrast with some of these. But again, there were other studies that show that actually, no, there isn't a response. So it's a strange one in terms of there's this variation in how bees are actually responding to the infection. But this was the first study that's ever been done in the UK doing this kind of work. And other studies have been done from different parts of the world. And it's possible that there's different strains of Varomorpha with different kind of genetic backgrounds and different diversity. And some of them may be more adapted to avoiding detection by the bees. It's also possible that the social context could be really important here. Remember, I used two bees to be real simple, but bees naturally live in group settings. And in the previous studies, they were introducing infected bees to groups of bees. So potentially the social context here is really important for triggering those social immune behaviors. And so to see whether that was the case, I then tested this in the colony in a way more realistic setting using observation hives. So here's a quick recap of what we know from the thesis so far. So we know that infection changes smell in the brood and the adults, but we failed to find any behavioral responses in pairs. So next question is, does the colony context matter? And so we had to go around setting up this field trial. And so I built these observation hives. These were two brood frames and a super frame. We'd have two introduction ports at the top so that I could introduce bees into the colonies and then watch their behavior with a feeder and an entrance and exit tube. And then you can see here, we had a specially made shed that I could place my observation hives inside. The university, for some reason, wouldn't let me put my colonies in the building. And so, yeah, we built this shed. You can see the bees happily using the observation hives there. So to try and isolate smell or olfaction as the mechanism underlying the behaviors in a field realistic setting, I had to find a way to perfume the bees with the smell of infection, but have the bee not actually be infected. And so we had these four different treatments. We had infected bees in exactly the same way as we've done previously. So at day 7 after the infection, these were our truly infected bees. And then this idea of infected smell, so bees that are perfumed with the smell of infected bees. So I placed 20 highly infected bees inside of a tube for an hour. They would rub off all of their chemicals and all of their volatiles, and they would deposit them inside the tube and then remove those bees and place an uninfected bee inside of it for 15 minutes, allow it to pick up the smells, and then introduce it straight into the colony and watch the behaviors to make sure that the perfuming process itself wasn't just, causing the differences. We did exactly the same thing, but we perfumed them with the smell of uninfected bees from that same colony. And then control bees, they were completely unmanipulated. They were just placed in clean tubes beforehand. And so the entire kind of experimental process here, as I kind of worked through it, I would first find infected colonies, make a lab culture of the spores so that we knew that we were using the same strain for all of the experiment. I then used two different colonies, either colony A or colony B. I'd collect the bees from the colony, rear them in the lab, and we would house them with wax from their original colony so that in the time it took in the lab, their smell didn't diverge too much from the original colony that was in the observation hive, so that when we reintroduced them, they didn't just smell very different because they'd been away from the lab. So the wax kind of kept them smelling like the original hive. Then we would either feed them with sucrose if they weren't to be truly infected or infect them, leave them for 7 days, and then do our introductions. So this is where we would either perfume them or just place them in clean tubes depending on their treatment. And so after I introduced them into the colony, I would observe their behavior. So I'd individually introduce them into each colony, up to 25 bees per treatment. And I'd do this on separate days. So our control bees would go first. So I'd do it one at a time, watch them for 5 minutes. And then once I'd introduced all 25, I'd then wait 30 minutes and then I'd repeat that. And then I'd do that for 5 more periods throughout the day. And then the next day, I'd do it with this treatment, next day with this treatment, so on and so forth. And I would be recording the frequency or the presence or absence of all of the different behaviors that they experienced. And so we could broadly categorize the behaviors I was tracking into these 7 different groups. We had aggressive behaviors. So these are things like biting, bees being dragged around the colony. You can see bees getting charged by the workers, by the donor colony bees. Things like avoidance behaviors. So sometimes a bee will go up and beg for food from another worker. They'll stick out their proboscis and beg for food, and the worker itself can either present them with a droplet of food, or they'll reject them and move away from them. So this is what we would count as avoidance behavior, as if their begging attempt was rejected. Things like antennation, self-explanatory. Bees would rub their antenna over each other, kind of smelling each other. Grooming behaviors. Social feeding would be trophallaxis, so sharing of food. Maintenance, whether the bee that was introduced was allowed to start looking after brood or kind of tending to wax cappings, and then whether they were just standard patrolling around the colony. So this is all of the behaviors that I recorded throughout this experiment as a percentage of the number of bees that were introduced. So say I introduced 10 bees and 5 of them were bitten, then 50% of them would have experienced an aggressive behavior. So we first looked at this really broad level of looking at things. And you can see for most of these ones, so social feeding, antennation, grooming, maintenance, and patrolling, there isn't really too much difference. And these are the times along here, so the different periods of observations. We have the introduction period and 1 to 5. But what's really interesting at this kind of broad level, if we look at aggression, both the infected bees and the bees that were just perfumed with infection, they weren't actually infected themselves, have a way higher level of aggression than the controls and the uninfected smell. Than these infected smell ones, they almost had 100% aggression rate in the introduction period. But this quickly tails off. After they've experienced that kind of initial level of aggression, it tails off and there's not really too much afterwards. Let me look at the avoidance behaviors, so whether their trophallaxis is being rejected or not. And it's, again, much higher in the infected and infected smell bees, almost at the same rate. But this one doesn't tail off over time, so they're consistently avoided throughout all of the time periods as I redid these observations throughout the day. So there's another way that we can look at these behaviors. So that was a very broad way of doing it. But I actually watched and recorded around 5,100 individual behavioral interactions. And in a similar way to the Vox, we can use ordination to understand these kind of changes in individual behaviors. So if we imagine that these bees here were bitten 7 times, and this bee here had 3 cases of trophallaxis, and it was very similar to these bees over here. Again, when we look at this in the space of this plot, they would cluster together because their overall behavioral profile are similar to each other. And then again, the ones that are very different would move into a different space. And each individual behavior will either push it in a kind of positive direction along the plot or a negative direction of the plot. And based on all the behaviors they experience, they'll move around these plots. And so this is what the plot looked like. As you can see that our infected bees and our infected smell bees, so the bees that are perfumed with infection, are very similar, even at the overall profile level. Of course, with bees and bee behavior, there's always going to be loads of variation. They rarely do what you tell them to do. But then our control and uninfected smell bees are pretty different. So if we look at actually up here in this quadrant, there's no single case of a control or bees that were perfumed with the smell of healthy bees in this section here. So all of these bees up here are just associated with either infected or infected smell bees. And so remember I said that we can look at how those behaviors push these points across, so whether they're contributing to where they place on the positive side of the axis or the negative side of the axis. So we can break down the individual contributions of each individual behavior. And the stars on this plot here just indicate that in my analysis, these were all statistically significant. But we can see that everything here associated with this side of the plot, so the positive side of PC1 along this way and the positive side of this axis here, are associated with these avoidance behaviors, all of the aggressive behaviors that we chose to record, so things like biting and chasing and dragging around the colony. And then interestingly, two of these behaviors here show that they significantly contributed to bees being placed in this section. So in other words, infected and infected smell bees were being groomed in this very extended and kind of it was a behavior I hadn't really seen before. They would be pinned down for almost the entire observation period and just excessively groomed the entire time. And so here's a couple of examples of those behaviors in the colony. So you can see this is what we would consider the extended dragging. So there's like a long period of a bee being dragged around the colony. So this was an infected bee from the experiment. And then this is an example of that extended grooming. So you can see, ignore this blue guy up here, but this poor lady here is being pinned down. You can see about the green tag there by about maybe 5 other bees, and they would stay there for the entire observation period. So it's interesting and different behavior, potentially, well, as this suggests, triggered by the bees being infected. But whether that's actually good for the colony or not remains unclear. So what we can say from this study is that bees that smell infected are treated as such. So reminder, these healthy bees were just smelled as if they were infected. They weren't truly infected. We found that aggression was kind of the initial response. It was very front loaded. It happens early, and then bees tend to switch towards avoiding them over time. And this could be an evolutionary adaptation. Aggression is quite fitness expensive. So it's very costly to be super aggressive within the colony all the time. It disrupts the natural processes. Whereas avoidance, if a bee comes up and begs for food and they smell weird, you can just avoid them. It's not too taxing for the colony. So it might be some evolutionary response. Interestingly, this extended grooming behavior, which hadn't really been seen before, could be that the bees are trying to groom the spores off of the infected bee to remove the spores, but it may also be another one of these strange manipulations by parasites. Because if I'm an infected bee and I'm covered in infectious spores, and I can encourage other bees to come and lick those spores off of me and potentially get infected themselves, that's a really good strategy. Whether that is the case, these are all just theories at this point, remains to be tested. But you could do a similar experiment to mine and then track what happens to those bees that are doing the grooming and then figure out whether they themselves become infected and see how their interactions change. What's also interesting from this is that we did manage to see the responses that were reported before in the colony and not in the lab pairing. So that idea that the social context is really important for social immunity has proven to be true. And so this tells us that smell is a crucial mechanism helping colonies assess infection risk. So that has a whole range of different implications. But we can't, using this study and this exact protocol, separate whether the volatiles were causing these behaviors or the cuticular hydrocarbons, because that perfuming process would have transmitted both the volatiles and the non-volatile chemical cues. But if we think back to the volatile study, at day 6 or 7, that's when the bee's volatile profile started to change. And there's previous studies that have looked at this infection and looked at their cuticular hydrocarbons, so their non-volatile signals, and they only start to change at around day 10. So that might support the idea that it is the volatiles that they're using. Though, again, this is a theory at this stage, but it does support it. So if we go back to the key questions of goals of the thesis, we've answered this question with the analysis that shows that honeybee brood produce volatiles associated with infection, and so do the adults. We've shown that it is specific to certain points of infection, and you can detect it as early as day 6. and that bees are using some form of olfaction to mediate their social immune responses. So bees that were perfumed, they just smelled like they were infected, were treated in exactly the same way as an actually infected bee. So what does this mean for bees? So if infection is as important for social immunity as we think it is, and we know how important social immunity is for bees, anything or any stressor that impairs their ability to smell could hinder their ability to resist disease. So we know a range of pesticides hinder bees' ability to learn smells and their memory. And a lot of environmental policy doesn't really look into sublethal endpoints. It says that you can use a certain pesticide or insecticide if a certain concentration of it doesn't kill bees and animals. But if you're using these and it's reducing their ability to smell, it could therefore reduce their ability to behaviorally avoid infection, which is really important for them, and therefore indirectly increase disease within the environment and within honeybee colonies. So understanding that smell is really important for social immunity could help us breed more resilient bees. There's a lot of work by Kyra Wagner at the moment looking into hygienic behavior and how we can breed more resistant bees or hygienic behavior in bees. So if we understand which mechanisms they're using in the adults as well, we could potentially target those behaviors in breeding programs if we know that certain receptors on their antenna can detect certain volatiles associated with infection, and then potentially in the future breed more resilient bees. But another important thing for this is it's not just limited to honeybees. Other social insects do this as well. So maybe bumblebees could also be experiencing this, and they would also be at the same threat from pesticides as well. So where do we go next? Well, in terms of the social immunity research, we would need to test whether bees actually physically detect these volatiles. It's all well and good saying that volatiles are different at day 6 and at day 12. But you can do studies where you plug kind of electrodes into the bee's antenna and you waft smells over them, and you can see whether they physically respond to those volatiles. But it's also important to understand how the volatiles and the non-volatile compounds are working together, because in real life, they're never going to be separate. It could be that the overall profile of volatiles and cuticular hydrocarbons are really important for how and what triggers different behaviors. So actually sampling both the volatiles and the cuticular hydrocarbons in the same experiment would be a really good next step. But also we could do similar behavioral studies to the one I described here with the observation hives and see if they're exposed, if that observation colony is exposed to pesticides, do they also show this social immunity response? Do they still remove the infected bees or maybe the bees that smell like they're infected? And also looking into other adult honeybee diseases. So obviously there's a range of different infections that infect adults. And so we could move on to kind of covering the broad range of different infections. In terms of disease surveillance, we could kind of look into whether the volatiles detected in this study can be detected in other colonies, in actually within the colony itself. So, like I was saying before, there weren't any unique biomarkers that we found here, but when we look at the overall profile, we could sample inside of the colony itself and do that with infected colonies versus uninfected colonies and see whether we can pick out the changes and the patterns in those volatiles. And that would really be the next step, fundamental step for starting to develop things like e-noses or gas sensors that can be used for beekeepers, which are much cheaper ways of monitoring a colony than things like PCR, much less labor intensive than doing the manual inspections yourself, and potentially could be used in the winter as well. If you just have your sensor in there, you don't then have to open it up in bad weather. And similarly, we could expand this kind of monitoring to other insect systems. So think of things like silkworm or feeder crickets as well. They're all susceptible to diseases and they're highly farmed. Using gas sensors like these could be really important if we can identify the volatiles associated with their infections. So if you'd like to read my actual thesis and kind of dig into the everything that I did here, it is publicly available. You can have a look. You can scan that QR code if you're interested. I now work at the Allerton Project, so I'm a senior scientist at Allerton. We do work on all sorts of regenerative agriculture. I personally do a lot of the insect research, but there's a lot of work on soil, and it's all about looking at how we can farm with nature using different kind of regenerative practices. And so if you're interested in hearing more about that, you can scan that QR code and sign up to the newsletter. And thank you all for listening, and all of the funders that helped me do this research, my supervisors, Carsten and Sarah, who taught me the science, and Peter, who taught me how to identify diseases, and Harold Williams, most of all. He's the beekeeper that I tracked down in Cardiff. He taught me everything I know about beekeeping because I had to know how to keep bees to do the research. And he's kind of my bee sensei throughout. So it wouldn't have been possible without Harold either. So thank you all for listening.
Thank you very much, Eamon. That's been really interesting, and we must obviously pass on our thanks to Howard for teaching you so well too. We'll have to get hold of him at some point. So we've got some questions coming in, apart from the obvious applause, which a few people are showing for you. Um, One of the questions is about your graphics that you use throughout,
Mhm.
and David in particular commented how great they are. And I was admiring them as well. And we were wondering if you created all of those yourself.
Some, it depends. So I used AI for a few of them and went back and forth with AI. Really handy. Some of them I drew myself. So these are AI, but you can probably tell which ones are going to be the ones that I drew. I drew this.
They're
um,
very good.
but
They're very
throughout,
good,
yeah,
and they're
you
very
can.
clear. So well done. I
Yeah,
think
thank you.
there's a hidden talent there. I'm surprised that you didn't use AI, since you've mentioned it, for analyzing some of the recordings that you made, because watching, I can't imagine how you maintained your focus for so long, even if it was only a 2 minute or a 5 minute or a
Yeah.
30-second count of behaviours. That must have been exhausting.
Oh yeah, yes, yeah, it absolutely was. Um, yeah, it's kind of just the struggle of a PhD, to be honest. But we did. I really wanted to use AI. Don't get me wrong. I didn't want to do that level of observation. Nobody should do that to themselves.
No.
But it's really hard actually to train AI on specific behaviors, and it's really tricky, especially in the context of a colony where the backgrounds can get quite messy. You can get propolis on the observation kind of perspex. Bees will disappear out of frame. Maybe they stick their head into a cell and they pop back up. And the AI has to be able to kind of identify all of these different factors. I did try initially, but it was just, in the end, it was just easier to do it myself.
Very good. I'm quite pleased to hear that in some ways. Deborah and Anthony have both raised a question about if bees are using smell, as you've indicated that they may well do. When we go into the hive with a smoker, or if
Mm.
we're treating them for varroa with thymol-based products, are we actually inadvertently then um, causing the bees more difficulty in sensing disease, or do you think they might get over those? I don't know.
So I think in terms of the using a smoker, I suppose at the time of using the smoker, it will disrupt their ability to smell because that's one of the reasons we use smokers, right? Because it
Hmm.
stops the alarm pheromone being sensed. But I mean, that will disperse. And when we're thinking about infection and these behaviors, the bees will go back to functioning normally after a while. I'm not sure with the different treatments, because if you think bees are going to be exposed to loads of different smells naturally, just throughout their natural behavior, there'll be bees coming back that are covered in floral volatiles because they're just absolutely covered in pollen. And so you'd think that they'd be able to kind of pick through that kind of noise if it's not something that is completely destroying their sense of smell. But potentially, some of the treatments that we do use could impact their ability to smell. It'd be interesting to find out.
Yeah, yeah, it would. Um, Paul's asking about the specific volatiles that are that you identified or that have been identified with Nosema itself, and what
Yeah.
sort of levels,
Yeah.
how?
So I you can go to my thesis to find out more if you want all of the details on that. So there were a few different ones that we found. I didn't dig into the kind of other analysis we did to draw out the volatiles in this paper, but they weren't bee pheromones. They were ones that were associated with kind of metabolism and gut microbes. And so there was one volatile that is associated with a certain type of microbe in the gut that can have antifungal properties. And so potentially, the gut microbiota itself is helping the bee defend itself from this gut infection. But
Okay.
I can't reel off the list of chemicals off the top of my head. I'm
Okay.
sorry.
We'll refer Paul back to your
Yeah,
document, your thesis, and he can follow that up for himself
yeah.
if he
I'll
wishes
be,
to. Thank you. And
Uh, Alison,
oh, hello.
it is Paul.
Hello, Paul.
Hello, Ayman.
hello, Paul.
What sort of levels were you detecting? Parts per million, parts per billion?
I think it was mainly parts per billion.
Okay. I'm,
Yeah.
I'm I'm just wondering. I'm interested in your electronic nose because I've used those things with Arduino, but they're very specific for gases, but
Mhm.
quite vague about levels. They do give you an indication, but that's all it is.
Yeah, so we didn't use electronic noses in these studies in particular. We were using gas chromatography mass
Yes.
spec. So yeah, so really sensitive stuff. I know with electronic noses, you kind of use a whole wide range of different sensors to try and pick up on these different patterns. I think it would take a lot more testing before the results that I've found can make it
Yes.
into an eNose. But there are some companies that are developing this for, I think, for Nosema at the moment, but also a range of other. If you've heard of AppySense, they,
Yes.
yeah, so they were quite interested in our research as well. But it is happening. It's in the process, but yeah, it's not something we in particular looked into that kind of transition from gas chromatography to sensor
No.
ourselves quite yet.
No, I can understand that. I believe there's some single chip mass spectrometers about these days, but I haven't used them.
Yeah,
I think that'd be the way to go.
yeah, potentially.
Thank you, Paul.
Thank you.
Okay. Yes, the gas noses seem really useful if they can be developed to sense other disease and pests as well.
Mhm.
But obviously something for the future. Our esteemed bee inspector Jack has asked, um, what was when you were doing the experiment with the two bees in the territory where they could either um,
So the ones in
yeah,
the arenas, yeah,
yes, in the arenas, that's the
yeah.
word. He was asking, what's the effective range for the bees to touch each other with their antenna? And does it require direct contact with the cuticle to detect cuticular pheromones? And if not,
Yeah.
he says, how close is close enough?
So generally, when you see bees antenating each other, it is incredible. They rub their antenna all over each other. I'm not sure. It depends on the cuticular hydrocarbon, and it gets into murky territory when you try to consider what a volatile really is. So generally, when we say volatile, it's stuff that's really moving throughout the air, but everything's a volatile if you heat it up enough. And so it could depend on the temperature of the bees as well. There's some studies that show that a bee returning after foraging has a different cuticular hydrocarbon profile to one that's not. So within our study itself, we controlled for that by just making sure that in our smell only condition, there was no space whatsoever for the cuticular hydrocarbons to be detected through antenation. But yeah, in terms of the effective range of cuticular hydrocarbons, generally it's touch, but
Mhm.
it could depend on which kind of the size of the cuticular hydrocarbon. The really long ones are really not very volatile, but I'm sure there's a few cuticular hydrocarbons under the right conditions that might be a bit more volatile, but nowhere near as long range as a truly volatile compound is.
Mm. I guess some of it depends on the sort of full, well, Jack was saying a colony level with the full cocktail of volatile and cuticular pheromones could the detection and response vary, perhaps as a consequence of the sort of background noise?
Absolutely. Yeah.
Yeah.
Yeah, for sure.
I guess also it might depend on the temperature because some compounds are going to be more
Mhm.
expressed freely when the weather's really warm, isn't it?
Yeah. So I'd wonder whether when you are developing these kinds of sensors, you almost need to consider the seasonal variation in volatility, right?
Mm.
So you'd need to have kind of uh, a trained gas sensor on a group of compounds that are robust to temperature differences as well. And I think the only way to do that is with more research.
Yeah, yeah. Now, Jack's made a couple of comments as well, which effectively support your idea of more research as well. But I'll just tell you what his comments are. So he says, you mentioned infectious transmission by induced grooming, and immediately CBPV comes to mind.
Mm.
Anecdotally, he thinks this is very likely, especially where the sticky nature of spores is considered. And he says, an elegant study to explore important questions regarding colony level immunity. Thank you, applause. And
Thank you
then
very much.
then he's got another question, which I think is he's obviously got this in mind that he would like to have this developed. And he says, with systems like Genie that can be used in the field, how feasible is an e-sensor? Could a gas MS fit in a briefcase?
Oh, I don't think you're getting it. Not to the level, I don't know, maybe one day. But I mean, RGCMS can barely fit in a room. So it's, but yeah, I'm sure as technology continues to advance, maybe one day we'll get there. And
Yeah,
especially with the developments in AI, even understanding the patterns and the analysis of it is going to get easier and easier as well.
yes, indeed. Um, okay, so I've got a question from one of our regular participants in these talks, Anthony and Anthony, who's based in America, and his question is about vaccinated queens, but we don't
Mm.
really have, well, in fact, I don't think we have any vaccinations permitted for bees in the UK, do we? So, but his
Think so.
question is, have you used your research approach and applied it to hives where AFB vaccinated queens have been introduced to assess the effectiveness of the vaccine? So I'm
No.
guessing,
So I think that is a great line of research for sure.
yeah,
And I would probably approach that question in a similar way, but not for us. We haven't
no,
done any of that.
you'd have to go to America to do that, basically, wouldn't you?
Yeah.
At the
If you've got
moment.
some funding, maybe
Yeah. Get Anthony to put his hand in his pocket. Um, Daniel's commenting, when smoking hives, seeing as smoke or molecules can stick to the bees maybe for a good period of time, how would this affect their smell? Would it inhibit their disease detection or for an extended period?
Yeah, so we kind of covered this before a little bit, but potentially you'd have to really test it in the colony, I think. I think it would be another one of those things where, I was saying you could do bees that are exposed to pesticides. You could also see how smoke is influencing it as well.
Mm.
But I would say that during all of my experiments, I had to smoke my bees to get them into the observation hives. So I was actively being a beekeeper around these
Mm.
colonies, and they still showed the behaviors.
Right. Yeah, that's interesting, isn't it? Um,
very much.
Thank you.
Very
Well
much.
done.
Great talk.
Thank you. Thank you very much.
Thank you very much.
Thank you.
So thanks very much, Eamon. I'm just going to
Thank
stop
you.
recording.